Es que no... ¿Sí? Ok. Muchas gracias a todos por asistir a este seminario. Espero que sea de vuestro interés. Esta vez hemos hecho algo muy breve, pero creo que muy intenso, muy interesante al menos. Hoy tenemos con nosotros a Guillaume Reveillon, a la mayoría de los, bueno, tanto los de la gente que estáis ahora conectados, lo conocéis porque es nuestro Field Application Specialist y es la persona que nos ayuda a nivel local con los equipos en las aplicaciones. Él nos hará una pequeña introducción sobre la imagen preclínica, hablará de aplicaciones en inmuno-oncología y además dará un pequeño repaso a nuestro portafolio de reactivos, que creo que es algo que es muy interesante. Luego también tenemos el gusto, el placer de tener con nosotros a Kevin Francis. Él es un gran entendido sobre todo en aplicaciones de enfermedades, en enfermedades infecciosas, pero además tiene un background muy importante. Yo creo que es un gran especialista que nos puede ayudar también. Tiene un elevado conocimiento de todo lo que es en la imagen preclínica. Entonces, bueno, yo no os voy a entretener. Yo soy Karma, para quien no me conozca, llevo la zona de Cataluña, Baleares, Galicia, Asturias y Cantabria a nivel comercial. Okay, thank you very much, Guillaume. If you want, you can start.
So good morning, everyone. Many thanks, Karma, for the introduction. Okay, so first of all, I would like to apologize for the French title, but I think you get the most point of it. So yeah, we're going to talk about all the different applications in immuno-oncology. So just the plan for today. So 3 different steps. So move. Okay, switch on, switch off. If I can see my presentation back, it would be nice. Okay, anyway, it's going to be in 3 different parts. The first part will be an introduction to in vivo imaging. Okay, then I'm going to talk about the application in immuno-oncology, and then I will finish by our, or I would say, re-engineered portfolio. So, first of all, why should we use in vivo imaging? Well, the answer is to be able to study biological mechanisms that are pathologic or not, right, in the animal. We can then assess therapeutic efficacy or safety of compound during after treatment. The idea of non-invasive imaging is to be able to image the same animals at different time points, and this will be fully aligned with the 3R principle, which tends to reduce, replace, and refine your experimental protocols. So we also have a good translation to the clinic because we are using animal models that are mimicking human pathology. So this is just an overview of all the different imaging modalities that you can find in the preclinical field as well as in the clinical field. Well, all of them are basically switched in 2 different main group, anatomical modalities, and on the other group, functional modality. On the anatomical one, you can find MRI as well as micro-CT and ultrasound. And in the functional one, it's divided in 2 with nuclear imaging and on the other side, optical imaging. For the nuclear one, you can find SPECT and PET, and for the optical one, fluorescence and bioluminescence. So here within PerkinElmer, we were using mainly 3 of them, micro-CT and optical imaging, but we have a new system which is coming in, an ultrasound system called the Vega. And the idea of this system is to be fully automatic, so there is no ultrasound expertise anymore. You will not have the transducer into your hand scanning the animal. So you just have to lay the animal on top of the system, and the system will scan the mouse. So, well, a different approach. The idea is to make ultrasound available for everyone, also for the people that don't have any expertise in ultrasound. So where is the signal coming from? It's coming from electromagnetic waves. They can have different energies or different wavelengths. It's 2 opposite units. So when you have the lowest energy, you have the highest wavelengths, and the opposite on the other side of the scale here. So basically, well, all of them are using electromagnetic waves, apart from ultrasound, which is using mechanical wave. For the visible range, so basically optical imaging, it's done between 400 nanometers and up to 800 nanometers. So it's called non-ionizing radiation. So it's completely free. So there is no dose delivered with this kind of modalities. If you want to be able to make some images, you need reporters as well as instruments. This is our instruments portfolio. So just an overview. We have many optical devices. Some of them are benchtop that can image 3 animals. Some of them are benchtop that can image 5 animals in one acquisition. And we have also systems that stand on the floor and that are able to do optical imaging in both 2D and 3D. One of them is, let's say, added with micro CT, so it's called the Spectrum CT. We also have a standalone unit called Quantum GX2, and the new entrance is the ultrasound system called the Vega. For the reporters, you can see here on the left the piece of DNA coding for luciferase. So this is for the bioluminescence approach. We need a substrate that is injected either IP or sub-Q, or sometimes IV, depending on the substrate. And on the right part, you can see fluorescence. It can be either using, again, a piece of DNA coding for a fluorescent protein, or it can be using a fluorescent dye, so semi-synthetic, that is grafted to a molecule of interest to do some kind of biodistribution studies. Okay, so far we have more than 16 publications with all the different platforms, and it can be found in any kind of research application, from neuroscience to oncology, cardiovascular diseases, and infectious diseases, which is going to be presented by Kevin. So among those 16,000 plus publications, we have a huge push on oncology. This is, I would say, what would make the technology used the most for. So many oncology applications, but over the years, we could see that we have some good percentage and increasing percentage in the other research field, such as, for example, infectious disease, as well as stem cells research and immuno-oncology, if you want to have a look at tumor cells. Now I'm going to change and going to move to the applications in immuno-oncology. First of all, it's mainly about being able to follow cell population. So in order to follow cell population, you need to tag them in order to track them. So the first strategy I would develop here is the use of a gene reporter imaging techniques, basically bioluminescence, using a gene coding for a luciferase. So the advantage are that you get a strong sensitivity, probably the most sensitive technique you can find today. So among all the different modalities I showed you before, MRI, ultrasound, and so on, this one is probably is the most sensitive. There is no way to go down to this number of cells with other modalities, whatever they are. So, well, the strongest sensitivity. It's also a viability marker. So all visible cells are emitting light. If they are dead, they are not emitting light anymore. It's long-lasting, so you can study the cells until they are dying. So you can follow them for months, for years. And you can see that also the piece of DNA will be transferred from the mother to the daughters. The concerns are more about the fact that we need to do a transfection, and this will, in the end, lead into clonal selection, because you will have to select the clone that is getting the piece of DNA, so your reporter, basically. So if you are working with heterogeneous pool of cells coming from a patient, then it's a bit more complex. You require a substrate that is injected to the mouse, and for some of them, we need cofactors, sometimes ATP, oxygen, sometimes just oxygen, depending on the luciferase you are using. So just an example showing the sensitivity. Here you can see that we can go down to really few cells if your cells are bright enough. We used to say that if you have bright cells, so emitting couples of thousands photons per second per cell, you could find 200 cells wherever they are localized, because here it's a bit more easy because it's just under the skin. So this is a different kind of luciferase you can use. So it's not exhaustive, not all of them are listed here, but you will find the major ones. Firefly luciferase, which is probably one of the commonly used luciferase, Renilla, Nanolook, and click beetle. This is I would say, the main ones that are used. Some of them are flash, so they are emitting a peak of light, and some of them are glow. They are emitting a light for a longer period of time. They also have different colors, so you can see here that the peak of those luciferases is not at the same wavelengths. And here I put you in this graphic in gray. This is the light absorption by hemoglobin. So we will try to avoid being inside this light absorption by hemoglobin. So basically we can see that we have an optical near-infrared window between 600, 650 up to 850 nanometer, where all the photons will cross in a way easier manner the soft tissue because you will see less light absorption. Okay, that's it for the different luciferases. Then you can also use another gene reporter. So you can use this time a fluorescent protein. Same strategy, you need to transfect your cells. But, well, the advantage are the fact that there is no substrate required. You can also use some other equipments like your fax or your microscope. It's also long-lasting, same as bioluminescence. And you can also see this transmission from the mother to the daughters. For the concerns, it's more about the transfection which is required, so same problem as bioluminescence. Another main concern is that it's not as sensitive as bioluminescence. So I have seen lots of research teams that they were working on bioluminescence and then they move to fluorescence and they don't achieve the same sensitivity and they are a bit disappointed. Compatibility of fluorescent protein with in vivo imaging is not very great because most of them are not in the near-infrared window. So most of the fluorescent protein are between the blue and up to orange, red. But when you are looking at really the near-infrared, there are just few families like IRFP, TurboFP, but apart from those 2-3 fluorescent proteins, all of them are in the bad range. So not very nice in order to be used in vivo. This is just a measurement of 3 different implantation, subcutaneously into the muscle and orthotopically because it's U87 glioblastoma cells. So just measured using 3D fluorescence. So you can see the X-ray to get the anatomical map, and we see the localization of those tumor cells in 3 different locations. There is another approach. If you don't want to use a gene reporter technique, we can also just incubate your cells of interest with a lipophilic dye. So we are just going to do the staining of the membrane, and this could be used in order to follow your cell population. So the downside is that, well, the good thing is that it's pretty easy to do. You just have to put your cells in touch with this agent for a couple of minutes, let's say half an hour incubation. So very easy, cost effective. We don't need to do any transfection. There is no need to inject any substrate. But the concern is mainly on how long you can follow your cells, because you cannot follow the cells for more than 3 days. So 3 days is really the maximum. After that, you don't really know what you are following. Is it the macrophages that have downgraded the cells that were dead? Well, we don't really know what is the fluorescence associated to. So there is no transmission to the daughters. So you will see also a kind of dilution of the signal over the division. And it's definitely not as sensitive as bioluminescence. But you can get good results. Here you can see those T cells that were just incubated with this lipophilic dye, in this case DIR. So fluorescence is emitted at 750, so very good at crossing soft tissue. And you can see the localization after IV injection. Early after injection, you can see some lymph nodes and some lymphoid organs. So basically the liver here, liver and spleen. Okay, then we can also target, well, we can also use our dye to graft those dyes onto antibodies. And the idea is to do kind of biodistribution studies, and you can also see if your antibody or your small peptide is targeting the tumor. So here we are using a synthetic molecule grafted with a dye at 680. And we are using this small molecule targeting integrins, alpha V beta 3 and alpha V beta 5. And we are using 4 different cancer models, so glioblastoma, colon carcinoma, and 2 lung carcinomas. And we have injected this probe into those animals. They were bearing the tumor here subcutaneously. It was injected on the right flank. And you can see over the different hours the distribution of the compound. So in all the different images, we did a kinetic for all the 4 different tumor models. And you can see between just after injection and up to 24 hours, the kinetic of light. And what we can see is that the signal-to-noise ratio between the intensity, the normal intensity, of the probe and the background is better at 24 hours after injection. You can see here, control lateral is a measurement which is done on the other flank of the mouse. Free cyanine 5.5 is just the dye alone without the small molecule targeting the tumors. And you can see that in both cases, we have the best signal-to-noise ratio 24 hours after injection. So this is the measurement we could get at 24 hours. You can see that from any kind of tumors, we have very good signal. And in the contralateral area, so just on the opposite flank of the mouse, there is a lot less, and same in the mouse with just the dye injected alone. Then you can collect all the different organs, and you can do biodistribution studies, organ per organ. So again, those ones are in French here, but you can find them in English here. So those ones were the one where we have injected U87 glioblastoma cells, just subcutaneously. And this is the distribution of just the dye alone. So you can see that just the dye or the dye linked to this semi-synthetic molecule targeting alpha V beta 3 integrins, you have a very different pattern. So this is the quantification that we performed on those organs. And again, the tumors are always showing the most intense fluorescence. And as you can see, in all the different organs, we have very low fluorescence. So we had a very good sensitivity here and a very good specificity. So which means that you can probably increase the dose. If you want to do some drug efficacy studies, we can probably inject more because we have very low amount of those products into the healthy organs. So this is all the organs. Well, you get the major signal from the tumor. Okay, what you can do is also to visualize those antibodies that were graphed with a dye in 3D. So here you can see the 3D distribution of the antibody, as well as the 3D localization of your tumor. So the red signal, 3D fluorescence coming from the antibody, and in blue is just 3D bioluminescence coming from the tumor. So in this case, we can clearly see the localization of the antibody. And again, we have a good targeting effect using our antibody. This is just a slide just to show you a multimodal approach. So it's the same animals, but we are using 3 different reporters. So you will see on the first one, we are looking at the host angiogenic response. It's bioluminescent images of transgenic mouse that are expressing luciferase whenever there is a synthesized, whenever VEGFR2 is synthesized. So you can see the angiogenic response here. And this angiogenic response is coming from the fact that we have injected tumor cells. So here you can see the tumor cells. So we could follow tumor growth using a fluorescent protein, bioluminescence to visualize the host angiogenic response. And here we use again an antibody which is graphed with a dye at 750 nanometer to see if we can target the tumor. And we can see a good target efficiency here using this antibody. So again, the same animals, and we have 3 outputs using 3 different reporters. Here it's also a multimodal approach. So in this case, we're using a model which is 2 different cell lines. One is called BT474, which is luciferase positive, and HER2 positive, and another breast cancer, MDA-MB231, which is HER2 negative. And in this case, we were using different approach to target those tumors. So you can see here the distribution of Herceptin, which is the trastuzumab graphed with zirconium. So we can see that only HER2 positive tumor is trapping this trastuzumab, which is targeting HER2. So here in the contralateral part, we see that MDA-MB231 is not detected. In optical imaging, we can also detect PET tracers. So this mouse was moved to the IVIS in order to do Cherenkov imaging and to detect the localization of the PET reporter here in a system which is not a PET. We also did 3D bioluminescence because those tumor cells were luciferase positive. So this is a 3D bioluminescence. And we also used another trastuzumab, but this one was fluorescent. It's called HER2-sense. And we can also clearly see the good targeting of the tumor expressing HER2 with this fluorescent antibody. Here I wanted to show you the impact of the use of T cells. So in this study, which is done in skin mice, we are using again glioblastoma, so U87, expressing EGFR. So those cells are luciferase positive, and they have been implanted in the right and left flank of those animals. And we did a couple of different treatments. The first treatment is nothing, no treatment, just PBS. Second one was using untransduced T cells, so they should not be that much effective. And the last one is using VEGFR-free CAR T cells. And in the case where we are using CAR T cells, we can see that the tumor growth is not going as quickly and as high as the one we can see in the control and the one using untreated T cells. So here we see between untransduced T cells and control, almost the same thing. We can see a normal tumor growth, and we can see a good therapeutic efficacy here using the CAR T cells. So we measured here bioluminescence, of course, and here the tumor volume. And we get good results with both measurements. So far we are having a look at the tumor cells. Here we are just having a look at the T cells. So we just performed a domiciliation study. So we took T cells, we transfected them with luciferase, and they were injected IV. And we were just checking where was the distribution of those CAR T cells. So as you can see here, they are mainly distributed in the spleen and in some lymph nodes. On this mouse, it's coming from the spectrum CT. So this is a real micro CT map coming from the mouse. And you can see the bioluminescent signal in 3D. But since we were not using a contrast agent to highlight the spleen, I just bring the spleen from a mouse atlas. So this is not the real spleen of the mouse. If we wanted to see it with micro-CT, it was needed to inject a contrast agent. Anyway, we can clearly see that we have a good colocalization between the T cells and the spleen. So here we are doing the same strategy, following cells, so following not CAR T cells, but following CAR M cells. So it's macrophages that are expressing the CAR. And well, in this study, they were just looking at for how long those cells are going to survive into the mouse. So it's just an healthy mouse. Yeah, this mouse has nothing. And CAR T cells was injected IV, but CAR M cells were injected IV. And we were just following the biodistribution of the cells. So where are they going and for how long they could survive. You can see that just after IV injection, they will be stuck in the lung and they will go in a second time in the liver and in the different lymphoid organs. And we can still see that up to 60 days after injection, we can still detect some cells, so they are not all dead. So now we are using those macrophages in order to see if we would have a therapeutic effect, again, tumor growth. So the model is using ovarian cancer, SCOV-free, injected IP, and 2 to 4 hours after the injection of SCOV-free IP, we are injecting same thing, either PBS, so nothing, untransduced macrophages, or macrophages that are expressing the CAR, so an anti-HER2. So 3 different groups, PBS, untransduced, and CAR-M. And here you can see the bioluminescent signal coming from the tumor cells, and we can see that we have a good survival curve after using the CAR-M cells directed against those tumor cells. In the case of the control, all the mouse were dead 13 days after induction. And in the case of untransduced macrophages, we have a small effect. So we can increase the survival for almost 2 times more, but all the mouse will be still dead before 16 days. So here you can see that some of the mouse are responder and some of the mouse are not responding. We can still slow down the tumor growth, but they will at some time die from it. Then we use another strategy. So here we are using VivoTrack, so one of our agents to label cells. So it's same approach as the one using DIR. So it's basically a lipophilic molecule that will stay into the membrane of the CAR-M cells. So here we are using those CAR-M cells tagged with VivoTrack in, again, 5 different models: esophageal cancer, gastric cancer, osteosarcoma, lung cancer, ovarian cancer. And we have injected the CAR-M cells, so 5-10-6, and 5 days after injection, we picked up the organs, so the tumor, the lung, the spleen, and the liver, and those 4 different organs. Well, tumor is not really an organ, but those 3 organs plus tumor was imaged, and in order just to see how much CAR-M was present in those organs. And here you can see the distribution with untransduced macrophages or CAR-M macrophages. We have a very similar distribution for both of them. You can see the blue part is the amount that is detected in the tumor. So we have a lot of them in the liver, but we have, let's say, around 25% that could be detected in the tumor of all the different tumor models. And we have a very clear difference in the gastric model, where we could increase by a lot the quantity of CAR-M compared to untransduced macrophages. So, well, the idea is that we can find those CAR cells in all the tumors from all the different models, with a big push on the gastric one. Here it's a nice study showing 2 different signals. So again, we are using CAR T cells in this case, and those CAR T cells were delivered differently. So we have 4 groups here. One group here using just control T cells, again, untransduced without any effect. We have T cells that have an effect and that are injected IV. Here another group with, again, T cells but injected intratumorally. And another one using T cells but delivered in a specific scaffold. And you will see the impact of this scaffold on tumor growth. So the first line here just show you the tumor at day 0. So we have implanted tumor cells. Those tumors were forming a mass. So you can see here the localization of the tumoral cells. Okay, perfect. On all the other ones, so the red lines, we can see the localization of the T cells. So you can see that at day 0, the T cells were, in this case, it's not T cells, it's just control cells. Well, they are just being injected IV, so they will go again through the lung, then they will go inside specific organs such as the liver and the spleen. But in this case, in the case of untransduced T cells, you will see that none of them will go in the tumor. So the tumor is somehow localized here. There is no signal at day 12 after injection, so no untransduced CAR T cell going to the tumor or targeting the tumor. Using IV injection, we can see that the T cell, again, they are making their distribution from the lung to the liver, and a good amount of them will be able to target the tumor, so we can find T cells starting from day 4 up to day 12. We can find some T cells colocalized with the tumor. And here we did another tumor measurement, 12 days, so at the end of the study, to see what was the growth rate between day 0 and 12 days. And you can see that when you are using untransduced T cells, we have a huge tumor growth. It's reduced using T cells injected IV, but it's way better using a local, well, using a local injection, it's almost the same as the IV one. But using a specific scaffold, which is composed of collagen and alginate, you will see that the cells will be able to expand way more, and the impact you will see on tumor growth is way higher than the impact you could see when it's directly injected into the tumor or IV injected. So here you can see they expand way more. So we have way more T cells expanding thanks to this material. Here I wanted to show you 2 different bioluminescent cell lines and 2 bioluminescent signals that are coming from the same animals. So here in this case, it was 2 different luciferase reporter, but it was using 2 different substrates. So one was using click bitter red and the other one was using nanolook. Here in this case, we are using the same substrate, infraluciferin, and two different mutants from firefly luciferase. So those two mutants were made by Clemens Lowick lab, and the idea of this mutant is to have two different peaks, but far enough so that we can use spectral mixing to separate the signals. So you can see that what they call the green firefly luciferase, it's not really green, because when you look at the wavelengths, it's almost 700 nanometer. And the other one, the red firefly luciferase, is emitting light at 740. So two luciferase reporters that are emitting light in the near-infrared range using a similar substrate. And thanks to our system and spectral mixing, we are able to visualize the localization of both cells directly into the mouse. So this is unmixed green, so the localization of the green signal. Here's the localization of the red signal. So the green signal is coming from the tumor cells. It was RegB lymphoma. And the red signal is coming from CAR T cells expressing CD19. On the first mouse, we can see that it's just the tumor alone without CAR T cells. And the second one is tumor plus CAR T cells that were co-administrated. So you can see here localization of the tumor cells. And in case of the one injected with CAR T cells, they are not growing as fast and as much as the tumor control. And here you can see the localization of the CAR T cells. So you can see a good correlation between the localization of the CAR T cells and the localization of the RegB lymphoma. Here you can see a composite image showing one reporter in green, so tumor cells, and in red, the T cells. And when they are fused, it turns yellow. So maybe you can see it, maybe not, but it's yellow here and here, here and here as well. So very nice studies. So this is not something commercially available, but it should, we try to tend to this kind of approach because when we are using other approaches, one of the luciferase reporter is emitting light in the blue or green range, which is not very sensitive. So this is a nice approach using two different luciferase reporter in the near-infrared range. So one of the last model, I think. Okay, again, using CAR T cells against PSMA. So we used here two different cell lines coming from the prostate. One is expressing PSMA, the other one was not expressing PSMA. So it's MIC-cap cells. And again, they were injected subcutaneously with or without CAR T cells. So here you can see MIC-cap, so the one expressing PSMA with no T cells. Here, there are PSMA negative tumor cells with the CAR T cells, and here the PSMA positive tumor cells with CAR T cells. And again, in terms of tumor growth, the fact that we are using CAR T cells can really reduce the growing rate of the tumor, which has a higher effect than the CAR T cells with PSMA negative cells, and higher effect than just the control cells. So here we are just looking at the tumor signal, and here we are looking at the T cell signal. So again, they did also kind of survival measurements of the T cells over the time to see how much T cells we have every day. And well, they are going down, but it's enough to induce an impact on tumor growth here. Okay, last slide for, let's say, the application in immuno-oncology. So this one is using CAR T cells that are overexpressing the c-Jun. And we can see that the c-Jun has a very huge impact on exhaustion resistance. So here we look at three different examples. MOC, so we have implanted tumor cells. We are using no CAR T cells, so this is a normal growth of the tumor. And after day 15, most of the mouse are dead. This is using the normal CAR T cells, so they are called 22BBZ, so expressing the bombzine receptor. So it's this kind of cells. So you can see that we have a good effect at the beginning of the treatment, but then due to exhaustion, there is no effect anymore. So just in order to avoid this exhaustion effect, so to resist it, overexpression of c-Jun have a huge impact. And here you can see the c-Jun overexpressing CAR T cells completely avoid exhaustion, and they have a very huge impact, which is lasting over the time. And here, those mice are almost cured from those NALM6 leukemia cells. So very, very huge impact of the c-Jun expression in your CAR T cells. Now I'm going to talk about all the different reagents. So we have, well, three main categories. Agents used for bioluminescence, they are called IVIS bright, and some of them are used in fluorescence, called IVIS sense. And we also have vessel view now, which is just microbubble that can be used for ultrasound. So just having a look at IVIS bright substrate, it's basically the substrate you can use with your luciferase reporter. So we have deluciferin, which is for click beetle and firefly luciferase, also for red efflux, so Luciola italica. And we are also selling celantervazine, which is a substrate for Gausia and for Renilla. And somehow we have also cell lines that are bioluminescent or bacteria. So just having a look at the different tumor cell lines we have, you can see that we are covering most of the major organs you can find in a mouse. So brain, breast cancer, colorectal cancer, fibrosarcoma, hepatic carcinomas. Well, you will always find a reference for any major organ. So all of them are at least bioluminescent using red efflux, but they could also be both bioluminescent and fluorescent. We have two cell lines like this, expressing dual reporters, so you can image them in both bioluminescence or fluorescence. Of course, in vivo, it's going to be way better using efflux because GFP cannot really be detected with a good sensitivity in vivo. We also have lentivirus particle. If you want to create your own cell line, you can use those lentivirus to make your cell line express luciferase. So we have different selection markers. One is using puromycin as a selection marker. The other one is using GFP, if you want to use FACS for cell sorting. We also have a lentivirus not using red efflux, but using Renilla, if you want to do dual imaging. This is available bacteria, so probably Kevin will talk a lot about them. He almost created all of them, I guess. So this is all the different bacteria that are nowadays available. Well, they're using the Lux operon for most of them, well, for all of them. We also have fluorescent agents. So this is the different agents that can be used in fluorescence. This kind of agent, the first category is called activatable agents. So those agents are quenched by default, and fluorescence is released whenever you have a specific protease, which is cleaving this dye. So we can use many different, let's say, agents in order to see presence of different proteases. Here I showed you two different examples. The first one here, so you can see the microcity map, you can see the surface topography of the mouse, so you can see two different tumors. Those tumors were bioluminescent positive, so we can see 3D bioluminescence here. So this is the localization of the tumor. And we have used two different agents co-injected together because they are using two different wavelengths. One is sensitive to metalloproteases. So you can see here the metalloproteases expression of your tumor. You can also see that they are trapped by the liver, as this is just the clearance of the probe. And the other probe is sensitive to cathepsins, so cathepsin B, cathepsin L, plasmin, so it's called ProSense. And here it's, again, you have the signal because those tumor cells are synthesizing cathepsins. We have another group of agents called targeted agent. In this case, we want to target a receptor, we want to target a specific, let's say, molecule in the cells. We can use them if you want to, for example, study apoptosis. So if you want to study proliferation, if you want to have a look at the FRAT receptor, if you want to see hypoxia. So many different receptors can be targeted. Here I just want to show you two different images done on the same animal. So it was a glioblastoma implanted in the right, part of the brain. And you can see two different signals that are, let's say, complementary. On this one, it's the use of IV-sense hypoxia. So we are targeting CA9, and you can see that CA9 is expressed in the middle of the tumor where we have an hypoxic core. And the other one was a vascular agent. So this agent was staying in the blood pool and is leaking in the tumor. according to the density of blood vessel you have in your mouse. So the funny thing is that you can clearly see a donut coming from the vascularized part of the tumor, and inside of it, in the middle, you can see the signal from hypoxia. So good complementarity between the two prods. And last category of agent called vascular agents. So those agents are passive agents. They don't target anything special, but they will stay in the blood pool and they will leak in two different cases. They will leak if you have new vessels, because those new vessels are fenestrated. So they will be able to leak and to accumulate inside the tumor. Or they will be also able to leak if you have an inflammation. So you will have a local vasodilatation. And then you will be able to, again, detect an accumulation of fluorescence. So we have different compounds with different sizes, so with different kinetic profile. Some of them can be clearly cleared from the mouse. Some of them will stay for a longer period of time, according to the size. Here is just a small study using Avastin as an anti-angiogenic approach. So this is just vehicle, so PBS was used. You can see tumor growth over the time. And here we have the vascularized part of the tumor. So more blood vessel density you have, higher signal you will detect. So in the case of the vehicle, we have a huge quantity of fluorescence that can be detected. And if you are using an anti-angiogenic approach, using Avastin, you can see that we are reducing the fluorescence. So blood vessel density is reduced, and we have a lower accumulation of fluorescence into those tumors. And I would like to thank you for your attention, and I'm ready for the question. Okay, let's see if you have some questions here in the chat box.
Do we have?
So far I could see any.
I will check. I will check.
At the end,
yes, we can answer at the end. Thank you very much, Guillaume. Now, please, if we have any questions, people from whom are online could write on the chat, and at the end of the session we will read. Okay, thank you.
So, good morning, everybody. So I'm going to follow on from Guillaume's presentation and talk about some of the applications in infectious diseases. So, Guillaume's already actually shown you these images previously, so I won't dwell on this. Let me move on. So, one of the major differences between, I'm talking mostly about infectious diseases, one of the major differences between using, looking at eukaryotic cells, looking at cancers, T cells, etc., is that we always tend to use the firefly construct. So normally the firefly construct or Ranella or nanoluke, etc., these are used in eukaryotic cells. In prokaryotic cells, one of the biggest advantages is that we can use a full Lux operon. This means that we have both the luciferase and the substrate incorporated into a single operon. So when you transform the cells, they can be constitutively bioluminescent without adding substrate. This means you can generate these types of images here without any substrate at all. This was generated at Imperial College in London. But what we can also do is, as Guillaume was saying, we can also combine this with fluorescent imaging. And what I'm going to do is go through and show you some examples that we've generated over the years. So just to start off by saying that there are a range of different, as Guillaume already said, a range of different luciferases that we can use. The most popular by far is firefly luciferase. This requires ATP and so it's energetic, metabolically active cells produce light. But if you use Ranella or nanoluke or Gaussier, then these are ATP independent molecules. So they're nice complementary molecules to use. Nanoluke in particular is incredibly bright, even though it's blue, it's incredibly bright and doesn't require ATP, and it's become a very popular molecule to use. What you can also do is you can combine the bioluminescent molecules with the fluorescent molecules using BRET. So you can channel energy, especially from the nanoluke, through the fluorescent protein to give you a further red-shifted wavelength, and I'm going to show you some examples of that later on. In addition, you can also, of course, use the system not only for Cherenkov imaging, but also for doing luminescent assays. Chemiluminescence can be used in the IVIS platform, as well as phosphorescence as well. So this is what you would see if you were imaging a plate. You can actually go into a darkroom with bacteria and see them by eye. So when I did my PhD back in the late 80s, early 90s, I used to just go into darkrooms and look at bioluminescence on plates using your eyes. But if you use just a standard camera, you can see that the cells are glowing without any substrate being added. This is all from one operon. The disadvantage is that these are very, very blue molecules. The peak luminescence of bacterial bioluminescence is 480 nanometers. This means that in the vast majority of cases, if you have an infection that's subcutaneous, you can see it very easily. When you start getting deeper, it becomes more challenging. Rule of thumb is that if you can cause an infection in an animal, you can see it. And that's because even though the signal is very blue, there's a very small amount of light. The photons in the red spectrum allow you to penetrate at depths as well. So even if you're looking at the lungs or the liver or the heart, you can still see that signal. It's massively attenuated. So I'm going to start off by showing you some old studies that we did. This is back from 2004, 2006. This was published in Science back then with Stanford University. So I engineered the strains, the Listeria strains, and then they were put into this mouse model. The reason I'm showing you this is because when these studies were run, with Listeria, you get a gastrointestinal infection, which normally only lasts 24, 48 hours. But in this case, Jonathan Hardy, who did this work at Stanford, instead of sacrificing the animals, simply carried on monitoring them. And because you've got this whole body imaging system, you can see novel phenotypes that come back. In this case, he found that the signal basically disappeared at day 3, but by day 4 was back in this very defined area in the animal. And it turned out that Listeria entered from the GI tract, entered into the blood, could move from the blood into the liver, and then drain into the bile, into the gallbladder. They then divided in the gallbladder using bile hydrolase, and then when the animal eats, you can see here, the bacteria move from the gallbladder back down into the intestines. And so you can actually show, it's like a Cox postulate, you can show you put the bacteria in, and then you can take them out several days later, showing that they are the bacteria that you put in, and that they basically move around in the animal over time. He even showed that, very weird phenotypes. They showed for one of the first times that the bacteria can go to the bone marrow. So they even studies showing that Listeria could go to the bone marrow. Of course, everybody knows it crosses the placenta, but you can do these studies. You can show that bacteria cross the placenta and infect the fetus as well. But one of the nicest things about using the bacteria being constitutive is that if you're doing just 2D imaging, you get this type of image here where you can see there's a signal somewhere in the, somebody in the lower quadrant of the animal. But if you're using 3D imaging, because you get a very nice consistent signal from the bacteria, you can see a very defined pattern. And in fact, what you can do is you can take these images, these movies, and you can link them together over time. So you make a 4D movie as well. So you can actually see tropism of the bacteria. This again is from Imperial College. This shows the cecal patch, the bacteria. This is a bacteria called Citrobacter odentium, and it infects a cecal patch in the large intestine and then spread throughout the animal or through the large intestine until it's excreted after about 12 days. You can also get some really nice defined images showing foci. So this is an E. coli K1 infection model where you can see in this rat pup dissemination of the bacteria to cause meningitis, crossing the blood-brain barrier and forming meningitis in the brain of this animal. So again, this is using the spectrum CT. But this is one of the biggest advantages of doing 3D imaging. So 2D imaging, we ran this study. This was one of the last studies we ran at our site in Alameda in California before it got closed down. Most of the research now is done in Hopkinton in Boston. What you can see here is this is a study where we had the mice were catheterized with uropathogenic E. coli. And what happens with this E. coli, when you catheterize the animal, the bacteria then ascend up the ureter into the kidneys to cause pyelonephritis. So you can see in this model, we ran 40 animals. And in this first group of animals here, about 20, well, it was actually 16 of the animals, you could see that they had this, what appeared to be true pyelonephritis. But then if we left them another week, virtually all of the animals had an infection, many with the same profile like this. But if we do 3D imaging, we can show that this is true pyelonephritis, this signal here. But this signal is actually where you've got a biofilm that's formed in the ureter ascending to the kidney, and that's why it's displaced. But if you look at the other mouse, this mouse here, at 3 weeks, it actually hasn't got a uropathogenic infection at all. It's not got an infection in the kidneys. It's got an infection in the gastrointestinal tract. And that's because the mice have eaten the food after they've urinated on it. So it's caused a GI infection. And the irony of this is that we were doing a treatment to treat a UTI infection. So when we treated these two groups of animals, all of these animals survived, all of these animals died because we were treating for the wrong disease. This here is a GI infection. We've also done I have a position at UCLA, and one of the things we do at UCLA, we do a lot of orthopedic infection studies. So when you're operating on patients, you tend to find a very low percentage. It's only a few percent, 2 or 3% of the patients end up having a post-operative infection. However, if you get this in your spine, it's very severe because you can't, you can debride the area, but you can't take the hardware back out of the patient because you can make them into a quadriplegic. So it's very difficult to treat these infections. So what we do is we generate these mouse models that mimic what you see in the clinic. So here we're putting a piece of Kirschner wire into the L4 of the spine, and then we infect it with bacteria. This is imaged in a spectrum CT. So what you can see here is that we've got the wire in the spine, and this is showing the bacterial infection. Most of it's in the soft tissue overlying the pin, but then there's also bacteria on the pin that have formed the biofilm. And then what you can see here is where we've got 2 different targeting dyes. Just like Guillaume was saying with the oncology studies, we've taken 2 different targeting agents, an antibody that specifically targets Staph aureus, and then we've taken vancomycin and labeled it with a near-infrared dye. And this was all done in collaboration with the University of Groningen. So we inject these dyes at the same time, these 2 different probes, and we can see the bacteria, and then we can see the different probes. And if you look at this on the film, you can see that here. So this is the antibody using a 680 nanometer fluorescent dye, and you can see the antibody then binds to the bacteria very specifically, whereas the vancomycin binds to the bacteria, but it also breaks down. After 3 days, it's 72 hours, or 48 hours here, the probe starts to break down, and so it doesn't target specifically, it's also non-specific as well. So what we've done over the years is we've used different methodologies for targeting the bacteria, and most of it's been using the Lux Operon. So when I first joined what was Xenogen then, this was back in 1998-99, I designed an operon that had Lux ABCDE, but one of the issues with this in gram-positives, it had polar effect. So I redesigned the operon, and what we did is we found out the bacteria were much brighter. But what we also did is I linked the operon so that it had a click beetle luciferase too. And you can see, this is what you're seeing with bacterial luciferase. This is what you see when you put luciferin in the animal and you turbocharge it. So you can also use the click beetle luciferase. The sensitivity increases by about 100-fold by using click beetle. So there's big advantages of using click beetle in bacteria as well. The caveat here is that when the bacteria become sessile, form a biofilm, this reporter doesn't work well. This one does. And so it's nice having a combination. So for acute models, this is wonderful. For sessile models, the bacterial luciferase works better. But it means you can actually then do really nice 3D reconstructions. We did this on one of the studies at Johns Hopkins, where you can see here that in this particular model, that can't be seen with bacterial luciferase, the bacteria have spread to the lymphatic system, and you can clearly define these 3 different foci with the bacteria in the lymphatic system by injecting with luciferin. And you can see again here the sagittal, coronal, and transaxial images using the spectrum CT. So I'm going to move on and show you some images also from using virology as well. So most of what I've done in my career has been bacteriology, but I've also dabbled in, I took my master's degree in virology, used to work for Wellcome Foundation back in the 80s, developing foot and mouth disease virus vaccines. So one of the things about viruses is some of the viruses are very small and they're hard to engineer. And so a way around this, and as you can see this from this paper from 2009, is to then use transgenic animals. So in this case, what they've done is they know that influenza causes cytokine storms, just like coronavirus causes upregulation of a lot of different cytokines. So if you take an animal, like this, in this case, interferon beta animal, labeled with luciferase, you can use pronuclear microinjection and generate a transgenic animal. That means when you've got upregulation of interferon caused by influenza, you can see it. So intuitively, you know these animals have got inflammation in the lungs, which is caused by the virus. You're not monitoring the virus directly, you monitor it indirectly by using this transgenic animals. This was before influenza was labeled. And then this is after influenza has been labeled. And the reason they now can label influenza is because they're using this very small luciferase called nanoluc. So nanoluc, very small, very bright, but very blue. But because it's so bright, it's about 100 times brighter than firefly, the smaller number of photons that you have can punch through, and you can then see the virus in the lungs of the animals directly. So this has been done with influenza, and it's also been done with coronavirus. This is SARS-CoV-2 work that was published just last year, showing that, again, taking SARS-CoV-2, labeling it with nanoluc, and showing that it can be imaged in this animal and even in the brain. This was done with using a spectrum. And we've also did this. I've also got a collaboration with the group at the Needle in Boston, and we published this work earlier this year, showing also that we can use this mouse atlas, so in the spectrum, to visualize expansion of this disease in the lungs of the animal as well. This is using a K18 mouse, which is positive for ACE2. But if you can't label, if you don't want to use a transgenic animal and you can't label your virus, there is a way around it. And this is a very clever study that was published in 2014. And what they've done here is they've taken a luciferase, but they've split it into the C and the N terminus. And in the middle, they've put a very unique catalytic enzymatic site. So this is a proteolytic site. So this proteolytic site is recognized by an enzyme produced by the virus. So when the virus infects the host, it produces this very unique enzyme, which then cleaves the luciferase. The 2 parts of the luciferase come back together, and you get a functional luciferase. And the nice thing about doing this is that you can do a stereotactic injection of this construct directly into the brain of the animal. And you can see that here, both the sham and then the infected animals. But only when the virus is present does it cleave the luciferase, and you get a functional luciferase. So again, the advantage of this is that you don't have to. So if you've got a lot of environmental viruses, patients coming in, and you want to be able to test it and see, is your drug specific against that? Just like with SARS-CoV-2, it kept evolving from the alpha to the delta to the Omicron variant, kept evolving all the time. You don't want to keep re-engineering the virus. What you can do is circumvent that by using an animal model such as this. And then one of the biggest issues has been over the years is that with firefly luciferase, people have wanted to image in larger animals. So a lot of the time with infectious disease, they want to go into primate models, go from, mouse models a lot of the time aren't very indicative of what you see in humans. They give you a good indication of the PK/PD of your drug, but if you're trying to actually get better, sensitivity for, say, for example, developing a vaccine, again, like SARS-CoV-2, you have to go into primates. So up to a few years ago, it was very difficult to get any depth of tissue. But then this Japanese group, this was published again in Science, this Japanese group, what they've done is they took firefly luciferase and they evolved it. And instead of just evolving the luciferase, they also evolve the substrate simultaneously, because it's like a lock and key, they have to fit together. And by doing that, they made this luciferase 100 times brighter than Firefly, standard Firefly. So it meant then that they could see this. In this case, this is looking at a marmoset. So they've taken adeno-associated virus, carrying this construct, and injected it into a marmoset. And if you go online, you can download this video yourself. This is from their paper. And this shows then being able to image in a much larger animal in real time. The thing about this is imaging in milliseconds. So you can see an awake animal being imaged. So if you wanted to do neurological studies, you don't have to anesthetize the animal. You can visualize this non-destructively, but of course the animal can stay alive for years with this construct inside it. And finally, what I'm going to do is show you some work that we've been doing at UCLA. This work was done at UCLA and also UC Davis as well. So what we're trying to do at the moment is we're trying to find ways of making nanoluke better to use in vivo. So this doesn't look very significant, but it is, because this is a construct that we designed, or I designed, where if you look at just nanoluke, this is what happens. You're taking different filters. So we're imaging in the IVUS system with an open filter, and this shows all the light being produced by that particular construct. This is on an agar plate, it's just E. coli. And what I've done is just dropped furimazine in the middle of the plate, the substrate, and then watching the light being produced. What I'm looking is at open, all the light being produced, all the photons, and then I'm looking with these different filters in place. And what I'm hoping to do is shift the wavelengths, because what I've done here is I've taken nanoluke and I've fused it to a long Stokes shift protein, which is a fluorescent protein. So the energy is moved from the nanoluke through the protein and shifting the wavelength. And you can see here that you're getting more photons. Only about 1% of the photons are being shifted. And it doesn't look significant, but if you're looking here, for example, at 670 nanometers, which is the optical window for seeing in animals, you got 20 times more light being produced by putting it through this fluorescent protein, which means you're going to get 20 times more light being produced in the animal if you use this construct as opposed to normal nanoluke. So it's actually a very significant difference. But the other beauty of this system is that this is a fusion protein, and it's also fluorescent as well. So it means if you're trying to do FACS sorting or tissue biopsies afterwards, if you want to do in vitro, in vivo, ex vivo, you can use the same reporter to do the whole continuum of this work. And not only that, what we've shown, so you can see this here, this is the equivalent of just a nanoluke construct. You can see the light very rapidly decays into the red spectrum. This is the optical window here. And the difference between here and here is you're getting about, on the order of about 50 times more light in most cases coming through. So it makes it much more sensitive. And finally, what we've done, it turns out this same protein, when you optimize it and put it into eukaryotic cells, and we're doing this again at the needle, we're looking at SARS-CoV-2 and looking at other alpha viruses, etc. When this same protein construct, this fusion, is expressed in eukaryotic cells, you get a huge peak shift. It's much more significant than in bacteria. And you can see that here, is that this second construct that I developed here has a massive peak shift in wavelength. So about 50% of the light now is shifted into the red spectrum. So this means this new nanoluke construct will give you much, much more sensitivity at depth inside the animals. But anyway, that gives you an idea of what we've been doing, and it shows you a lot of the bacterial work. I was a lot faster than Guillaume, but so he gave you most of the basis here. So if there's any questions, then please go ahead and ask.
Thank you very much. We need to double check if we have any questions from the audience. No. Yeah. Si alguno de vosotros tenéis preguntas, podéis hacerlas, ¿eh? Parece que no. Here we have one question. They are asking for, okay, they say good sensitivity in subcutaneous tumor models, but what about metastasis? For example, lung metastasis after injection of tumor cells.
Do you want me to answer that? So when it comes to looking at metastases, as Guillaume showed you, you can detect very small clusters of cancer cells. But as I also showed in that model with the marmoset, is that you've now got the ability to see single cells at depths in tissue. So the paper that Guillaume showed was using T cells. There was a paper we published in 2010 showing you can see a single cancer cell subcutaneously. Since then, people have shown that you can, the group from Japan showed that you can see single cells at depth in larger animals as well. So if you're looking at metastases, if you have a primary tumor and then you get metastatic lesions forming after the fact, in mouse models, you can easily see them, just a few cells, just a very small cluster, or even theoretically down to one single cell. So yes, it all comes down to a combination of expression of the luciferase within the cells and the energetics, how much ATP is being produced when you've got a biologically active system, such as using Firefly. If you're using Gaussian or you're using Nanoluke, you don't need activity, ATP, so you can see sessile cells. But in this particular case, if you're looking at metabolically active metastases, Firefly or Akaluke, the Japanese version is very, very bright, and you can see very small numbers of cells. Okay. Any other questions? Nope. Okay.
Well, quick question. So, talking about RET, I think Antares is one of the most well-known RET. So, Nanoluke compared to Enroll or something. Mm-hmm. So, how is behaving your combination between Nanoluke and your fluorescent protein compared to, let's say, the gold standard Antares?
One of the things is what I noticed that the peak bioluminescent signal from Nanoluke is 460 nanometers. So if you're using a red-shifted protein already, if you're using something like cherry or plum or tomato or whatever, you're already up at above 500 nanometers. You're up at like 540, 560 nanometers, and the amount of energy that can be transferred from Nanoluke to that, you're only talking about 1%. But if you're using a long Stoke shift protein where you excite in the same wavelengths as Nanoluke, and then you emit up in the red, the energy that you transfer is massive in comparison. And so it was amazing to me that people kept taking, red fluorescent protein or derivative red fluorescent protein rather than taking something that's actually exciting at the peak wavelengths of Nanoluke. So that's why I chose that construct. And it seems to work a lot better. So, but we haven't published any of this work yet, so we've still got to publish it.
Which equipment is used for the marmoset?
Sorry?
Which equipment would be used for the marmoset?
That was actually using an EMCCD. So we actually used to sell one of those cameras. So you can get, our systems use integrating CCDs. They're back illuminated. So basically what they do is they accumulate the signal over time. So as you capture the image, rather than keep showing it in real time, they accumulate the image over time and show you the final image. And that's the most sensitive way of collecting data. However, because they had so much light being produced, they use an ICCD or they used an EMCCD. I think it was actually an EMCCD, which is an electromultiplying CCD system. So by doing that, they've got a camera that continuously gives you a readout. It's not quite as sensitive, but they're still pretty good. So they would have used that camera on this. So it's very similar to our cameras, but it's basically putting the marmoset in a dark room and using this camera externally. So that can be done. The army, both in the UK and in the US, use these type of cameras when they're imaging things like goats. When they're doing horrendous models on larger animals, they tend to use these external cameras. So you can use those external cameras. The disadvantage is you can't get absolute quantification from them. So you can get an indication of what's happening, but you can't get quantification because they're not calibrated correctly. So, but with larger animals, you could put that animal into our system, and they have done that actually. In the paper, they put it into our system as well. So a marmoset will fit in our system, and African green monkey will fit in the system as well. But when you want to do real live imaging, you need a kinetic camera, and that would be an ICCD or an EMCCD camera.
It was not an experimental equipment. It was not an spectrum CT.
No, it wasn't doing that. It was using a separate setup in the paper, yeah, because they wanted to do real-time imaging. We used to sell a system called the Kinetic, which had the same camera, but nobody bought it, so we stopped selling it. But it's not. It's actually quite easy. It'd be quite easy to unbolt one camera and put the other camera on, and then it would work like an ICCD or an EMCCD. But the reason I was showing that image was I wanted to show you what's possible using the luciferase. So the luciferase is so bright, it's to show you what's possible even in a large animal using biophotonic imaging.
Okay. If there is no other question, because I think that there is no one from here, but en cualquier caso, si alguien de los asistentes online cree que tiene alguna pregunta que le pueda ocurrir, pues se le puede ocurrir posteriormente, tenéis nuestros contactos, al menos el mío seguro, porque es el que, bueno, ha hecho la difusión del evento. Entonces, bueno, nos las podéis mandar perfectamente sin ningún problema y las podemos ir resolviendo. Bueno, daros las gracias a los que habéis venido y a los que estáis online, a nuestros ponentes. Muchísimas gracias. Thank you very much, Guillaume. Thank you very much, Kevin. Y nada, dijimos que íbamos a ser breves y así ha sido. Muchas gracias.